Hello guys, good afternoon. You are welcome to my YouTube channel, the Explicit Tutorials. In today's video, we shall be treating a topic in biology called digestive system. Alright, digestive system means the various organs or tissues or systems that are responsible for what? The process of digestion, okay? If you are asked the meaning of digestion, is different from the meaning of a digestive system. All right, they are the organs or the systems that carry out what? Digestion. What about digestion? Digestion means what? The process whereby larger food molecules are broken down into simpler forms. It is the process whereby larger food molecules are broken down into simpler particles or into simpler forms under the influence of enzymes and hormones, all right, for absorption and what? Assimilation. Now, I just said two terms, absorption and assimilation. Now, JAMB, it is very important you know the difference between absorption and what? Assimilation. Now, I said that digestion means what? The process whereby larger food molecules are broken down under the influence of what? Larger food molecules are broken down into simpler forms or particles under the influence of enzymes and what? Hormones for absorption, or which can be absorbed into the bloodstream and therefore is what? Assimilated. Now, absorption and assimilation, they are two different words. Absorption means the process whereby the digested nutrients are stored in the bloodstream. All right, they are stored in the bloodstream. That is what? Absorption. Now, the process whereby these nutrients are converted into energy for work. When these nutrients are converted into energy for work, you could call it what? Assimilation. But when these nutrients are absorbed, all right, into the bloodstream, you call that what? Absorption. Please take note of that. Now, absorption involves several processes. If I don't call it digestive system, I'll call it what? Alimentary canal. All right, if I don't call it alimentary canal, I'll also call it what? Gastrointestinal tract. Gastrointestinal tract. Now, what are the various alimentary canals? One, you have the mouth. Now, before I take number two, let's discuss what the mouth is all about. You know, the mouth is made up of the tooth, and the mouth is also made up of the tongue, and the mouth also has what? Salivary gland. All right, and the mouth is also made up of the soft and hard palate. All right, it's made up of the soft and hard palate. The soft part is the lower part of the mouth, while the hard palate is the upper part of what? The mouth. Now, the mouth has the tooth, the tongue, salivary gland, and what? Soft and hard palate. Now, the tooth is meant for what? Grinding. It's meant for cutting and what? Grinding. Please take note, in herbivores like rabbits, all right, rabbit has 28 sets of what? Tooth. 28 sets of tooth, and they have incisor. All right, they don't have canine. Please, they do not have canine. They have premolar, and they have what? Molar. Now, going to the fact that canine is absent. All right, the space between the last set of incisor and the first set of premolar, you have a space called what? Diastema. All right, diastema is that space found between the last set of the incisor and the first set of what? The premolar. Diastema, which means that diastema is replacing canine. Canine is meant for what? Tearing of flesh. Okay, therefore they do not have the ability to what? Tear flesh, because canine is absent and it has been replaced by a space called what? Diastema. Diastema is found in herbivores. A good example is what? Rabbit. Rabbit has 28 what? Set of teeth. Now, the tongue. All right, tongue. Now, what do we know about the tongue? Now, the tongue has a sensitive cell present in it. The tongue has a sensitive cell. Now, these sensitive cells that give the tongue to taste chemical substances are called what? Taste buds. Taste buds. Taste buds are sensitive cells that help the tongue to taste what? Chemical substances. Now, we have different types of what? Taste. We have salty taste, we have sweet taste, we have sour taste, and we have a salty taste. Now, listen, very important. Sweet taste, please take note. Sweet taste is detected at the tip of the tongue. Sweet taste is detected at the tip of what? The tongue. Now, bitter taste is felt at the back of the tongue, or you say at the base. At the base of the tongue, you have what? Bitter taste. Now, you now have sour and salty. If is just by the side. Please take note. Sweet taste is felt at the tip of the tongue. Bitter taste is felt at the back or the base of the tongue. Then sour and salty taste are felt at the side of the tongue. Take note of that. The next one is what? The salivary gland. Now, the salivary gland is made up of other glands. All right, let me rub out this very well, please. The salivary gland is made up of other glands. These glands are the parotid gland, that is the constituent of the salivary gland. We have the sublingual gland, and we have a submandibular gland. Now, these are the various salivary glands, all right, or the various glands that make up what? Salivary gland. All these glands, their primary aim is to secrete what? Saliva. Is to secrete saliva. Now, saliva is not a waste product. I did not say excrete. I said what? Secrete. Now, secretion is when useful substances are produced in the body, while excretion is when useless substances are excreted from the body, or they are taken, or they are, okay, what should I use now? Okay. They are discharged. Good. They are discharged from the body. But in secretion, the substances secreted, they are useful. All right. The body makes use of the substances secreted through what? Secretion. Now, I said the primary aim of these glands is to what? Secrete saliva. Hope it's recording. Excuse me. Yes, it's recording. Sorry for that. The primary aim of these glands, I said, is to secrete saliva. Now, the saliva has a type of carbohydrate digestive enzyme. The saliva has a type of digestive enzyme called what? Tyalin. I recap. The saliva has a carbohydrate digestive enzyme called what? Tyalin. Please, the tyalin is what? An alkaline substance. Take note of that. It is not acidic. It is what? An alkaline what? Substance. Now note, digestion of carbohydrates starts from the mouth. Digestion of carbohydrates starts from the mouth. Now the function of this tyalin present in the saliva, it helps in bringing the food together in the form of a solid mass called bolus. And recap. This enzyme present in the saliva helps in bringing the food molecules together in the form of a solid mass called what? Bolus. Now I can say that the partially digested food in the mouth is called bolus. The partially digested food in the mouth is called what? Bolus. So I've explained everything that occurs in the mouth. I said the mouth has the tooth, tongue, salivary gland, hard and soft what? Palate. So take note, diastema is found in herbivores. A good example is what? Rabbits. Alright, diastema replaces canine. Therefore, they do not have the ability to tear flesh, okay? Take note of that. And of course, carnivores, okay, they have canine, with which they use to get hold to their prey. Is for tearing of what? Fleshes. Take note of that. And the next one is the pharynx. Number 2 is pharynx. And the pharynx is just that tube that joins the mouth to the esophagus. All right. It is the tube that joins the mouth to the what? Esophagus. That's what? The pharynx. Now, the esophagus does not play, that is, does not play a wide role in the body. Okay, so it's just meant for the what? The passage of food. Now, as soon as the bolus is swallowed, it passes through the esophagus or gullet. I don't call it esophagus, I call it what? Gullet. Now, the movement of food in the esophagus or gullet is called what? Peristalsis. The movement of food in the gullet or esophagus is called what? Peristalsis. Now, what is this food moving? Bolus. Bolus, when it moves through the esophagus by the contraction and elevation of it, you call it what? Peristalsis. Take note of that. Now, the fourth one is the stomach. Now the stomach has an upper part. The stomach has an upper part called the cardiac sphincter. The upper part of the stomach is called what? Cardiac sphincter. The lower part is called what? Pyloric sphincter. All right. Pyloric sphincter is what? The lower part of the stomach. The cardiac sphincter is the upper part of what? The stomach. Of course, you don't need an oracle to tell you that the stomach is highly acidic. The stomach is what? Highly acidic. And please, carbohydrate is digested in alkaline medium, never in acidic what? Medium. Now, as soon as the food enters the esophagus, as it's coming, the cardiac sphincter opens for it to enter into the stomach. Please take note. As soon as the bolus gets to the stomach, the first thing the stomach does is to neutralize the alkaline content present in the bolus. Now, the bolus, I said, is a solid mass that is formed by this word thiolane. It contains alkaline content. As soon as it gets to the stomach, the first thing that happens is that the stomach neutralizes the alkaline content present in meat. Take note of that. The stomach also produces a concentrated acid called what? Hydrochloric acid. The stomach produces a type of acid that is concentrated. You call it what? Hydrochloric acid. Now, the stomach is actually triggered by what is called what? Gastric gland. Gastric gland. Now, this gastric gland produces what? Gastric juice. All right. Gastric gland produces what? Gastric juice. Please take note. Gastric juice is made up of strepsin and trypsin. Take note of that. This gastric juice is made up of strepsin and what? Trypsin. Take note of that. Now, as soon as this HCl is produced by the stomach, what it does is to convert pepsinogen to pepsin. The function of the HCl produced is to convert pepsinogen to what? Pepsin. It therefore means that pepsinogen is what? The inactive form of pepsin. Then pepsin is what? The active form of what? Pepsinogen. The function of this pepsin is to convert protein to peptone. The function of the pepsinogen is to convert protein to what? Peptone. That is the function of the pepsin rather. The function of the pepsin is to convert protein to what? Peptone. Take note of that. Please, they'll ask you which of the following is digested in the stomach. Which of the following is digested in the stomach? They'll give you option A, carbohydrate and protein. B, protein only. C, protein and fat. D, fat only. Please, the answer is what? Protein only. Because protein is best digested in acidic medium and that is what? The stomach. Okay. Now I said pepsin converts protein to what? Peptone. HCl convert pepsinogen to what? Pepsin. Please, a type of enzyme, okay, found in the stomach called renin. The renin helps to curdle milk. All right, it helps to curdle milk. Okay, I think should I above? Okay, let me above this part. Now the renin, I said, is found in the stomach. What does it do? It helps to what? Curdle or coagulate milk. All right. So which of these curdles milk? It is renin. That is RENNIN. Take note of that. So moving from the stomach, we now have the small intestine. I believe what I've said so far is very, very understood. So I said protein is converted to peptone by pepsin. HCl convert pepsinogen to pepsin. And I said the stomach has what is called gastric gland, which produces what? Gastric juice. Gastric juice is made up of what? Strepsin and what? Trypsin. Take note of that. Now moving from this, okay. As soon as the food gets to the stomach, there's what we call trituration. Trituration is a phenomenon characterized by the churning of food molecules in the stomach. All right, churning. Now, when the muscles of the stomach contracts, there's what? Grinding of food molecules. Now, this grinding process that occurs in the stomach is through what? Trituration, and is by what? Churning. The muscular contraction of the stomach helps in breaking down food molecules through trituration. Please take note of this. The partially digested food in the stomach, all right, the partially digested food in the stomach is called what? Chyme. The food molecules that have been partially digested in the stomach is called what? Chyme. Say chyme. Now chyme is to the stomach, chyle is to the intestine, then bolus is to the mouth. The partially digested food in the mouth is called bolus. Then the partially digested food in the stomach is called chyme, while the partially digested food in the intestine is called what? Chyle. Take note of that. Now, as soon as the food molecules are digested or are broken down already, the pyloric sphincter opens. The food moves into the small intestine. All right. So, before I think something just ran into my brain now. Oh, I can't remember again. So. Okay, maybe before I round off the class, I'll remember it. So, okay, the stomach. All right, the small intestine is made up of 3 parts. The small intestine is made up of 3 parts. All right, we have the duodenum, and so we have the ileum, and we have 3, the jejunum. Now, these are the 3 subdivisions of the small intestine. Good, I just remembered. Now, man has a well-defined stomach. Now, please, birds have beak. Now, move from the mouth of birds. The mouth of birds is modified into what? Beak. Now, the food moves from the mouth to the other part called crop. All right, crop is where food is stored temporarily. All right, food is stored temporarily in the stomach. Moving from the stomach, you have what is called gizzard. Now, this is where my emphasis is. The gizzard is where is a part that contains stones and other tiny particles. What does the gizzard do? The stones present in the gizzard help in breaking down the food molecules. Please, the gizzard of birds, all right, help. Even earthworm has gizzards. The gizzard of earthworm and birds contain stones and other smaller particles that help in what? The mechanical breaking down of what? Food molecules. Take note of that. Then also permit to say this: ruminant animals, of course, they are animals that have what? Four stomach. All right, you can also call them polygastric organisms. Polygastric organisms. Poly, they have four stomach compartments. The first one is what? Rumen. Two, reticulum. Reticulum. Three, omasum. And four, abomasum. These are the four stomach compartments present in ruminant animals, which are otherwise called polygastric animals. Rumen, reticulum, omasum, and what? Abomasum. Good. My emphasis is based on rumen, which is the first stomach. Now, when humans ingest vegetables, after absorption, going to the toilet at any time, you will observe that you are excreting out same vegetables that you've eaten before, because you do not have cellulose digestive enzyme. But in animals, grazers, all right, they have enzymes, all right, that help in digesting cellulose. You know that the cell wall of plants is made up of a type of polysaccharide called what? Cellulose. Now, ruminant animals, cellulose is present in the rumen. Now, this cellulose present in the rumen, there are bacteria present, or there are bacteria inside this rumen. The function of this bacteria is to help to break down the cellulose present in the vegetables. Therefore, the feeding association between the bacteria and rumen of ruminant animal is what? Symbiosis. The feeding association is called what? Symbiosis, because the bacteria helps the bacteria help in breaking down cellulose, while the rumen provides shelter or protection for the bacteria. Take note of that. Then, as soon as the food and cellulose is being digested in the rumen, what happens? It enters into the reticulum. How should I put it now? As soon as they are resting, the food moves to the reticulum. Then after which they bring out the food again to be rechewed the second time. All right. This food that has been brought out the second time is called what? Cod. This food that has been brought out by this ruminant animal is called what? Cod. The process by these food molecules are brought out is called what? Regurgitation. Regurgitation. All right. Take note of that. They bring out their food molecules through a process called what? Regurgitation, to be rechewed the second time. Take note of that. So these are the first stomach compartments of ruminant animal. Ruminant animal has the first stomach called the rumen. That is where you have bacteria that digest what? Cellulose. All right. Take note of that. Now this cellulose is broken down into fatty acid and glycerol in the rumen. Take note of that. They'll ask you, which of the following stomach compartments ruminant animals are cellulose digestive bacteria found? First, stomach, ruminant animals. All right. Now they have the ability to regurgitate. All right, and the food molecules they bring out to chew again is called what? Cod. Cod. C-U-D. Cod. Now, where is the board paper, please? Okay. Now, let's come back to duodenum. All right. Now, in birds, I just remember something. In birds, the parts of their alimentary canal that play the role of stomach in higher animals is called proventriculus. I recap. The proventriculus in birds plays the role of stomach in higher animals because birds do not have stomach. They have a proventriculus. It is this proventriculus that does the work of stomach in higher animals. I said crop is that region where food molecules are stored temporarily. I said gizzard contains stones and other tiny particles that help in breaking down food molecules. Now, the duodenum of the small intestine, this digestion occurs in the duodenum. Now, the duodenum has an enzyme, or it produces an enzyme. Okay, let me put it like this. The gland found in the stomach is called what? Pancreas. The type of gland present in the stomach is called what? Pancreas. Now, this pancreas will ask you which of the following has the ability to produce both hormones and enzyme. It is what? Pancreas. Now, as hormone, it produces what? Insulin and what? Glucagon. Then, as enzyme, it produces thrombokinase or kinase. The insulin helps in converting glucose to glycogen, and the glucagon converts glycogen back to what? Glucose, resulting to what is called diabetes what? Mellitus. Now, the thrombokinase, I said this pancreas is found in the loop of the duodenum. Now, this thrombokinase produced by the pancreas in the duodenum converts trypsinogen into what? Trypsin. The function of the trypsin is to convert peptone to polypeptide. Now, the polypeptide is converted to what? Amino acid by an enzyme called what? Erepsin. The pancreas present in the loop of the duodenum has the ability to produce enzymes and hormones. As hormones, it produces insulin and glucagon. Then the enzyme it produces is called what? Thrombokinase. The function of the thrombokinase is to convert trypsinogen to what? Trypsin. This trypsin, on the other hand, converts peptone to polypeptide. Polypeptide is converted to amino acid by what? The erepsin. That is for that. And the end products. Now, I told us just now that protein is converted to what? Polypeptone by what? Pepsin. Now, JAMB or any WAEC candidate, they'll ask you which of the following best expresses the sequence of protein digestion. Which of these best expresses the sequence of what? Protein digestion. Protein to peptone, peptone to polypeptide, polypeptide to what? Amino acid. So if you are asked what is the end product of protein, you say what? Amino acid. What are the monomers of protein? You say amino acids. What is the polymer of amino acids? You say what? Protein. Now, protein in the presence of pepsin is converted to what? Peptone. Peptone in the presence of trypsin, that is this word trypsin, is converted to what? Polypeptide. Polypeptide in the presence of erepsin is converted to what? Amino acid. Take note of that. Digestion occurs in the duodenum. Now, please, I just remember something. The folds present in the stomach, which help in the grinding or which gives it large surface area for what? Digestion, is called rugae. Rugae are folds found in the stomach. What do? They help to give it large surface area for what? The breaking down of food molecules. All right, take note of that. Now, the next one is ileum. Which of these part of the small intestine do digestion and absorption occur? Please, digestion and absorption occurs in the ileum of the small intestine. Now, they are the finger-like projections found in the small intestine is called what? Villi. All right. The thread-like structures found in the small intestine is called what? Villi. The function of the villi is to provide what? Large surface area for what? Absorption of food molecules. All right. Take note of that. The villi are finger-like projections found in the small intestine. What do? They help in providing larger surface area for what? Absorption. Now, in the small intestine, you also have what is called thread-like structures called succus entericus. Succus entericus. Now, there are 5 enzymes present in the small intestine. 1, we have lactase. Okay, let me bring it up, please. The enzymes found in the small intestine are 1, we have lactase. 2, we have maltase. And we have 3, sucrase. 4, trypsin. And 5, erepsin. All of these are found in the small intestine. What do they perform in the body? Now listen very well. Lactase is an enzyme that converts lactose to into glucose and galactose, which means that glucose plus galactose will produce what? Lactose. Now, maltase converts maltose to 2 molecules of what? Glucose. So I'll say glucose plus glucose, and we have what? Maltose. Now, sucrase is an enzyme that converts sucrose to glucose and what? Fructose. Which means that glucose plus fructose will give us sucrose. And please, lactose, maltose, and sucrose are all disaccharides. Disaccharides are formed when 2 or more monosaccharides are joined together by the elimination of what? Water molecule. Now, examples of monosaccharides are glucose is a monosaccharide, galactose is a monosaccharide, all right, fructose is a monosaccharide, glyceraldehyde is a monosaccharide, all right, arabinose is a monosaccharide, ribose is a monosaccharide. All of these are monosaccharides. When 2 or more monosaccharides are joined together by the elimination of water, you get what? Disaccharides. Okay, now this is what I'm trying to say. Monosaccharides, they have general formulas C6H12O6 plus another monosaccharide C6H12O6. I'm going to eliminate water. That is minus what? H2O. C6 plus C6, I have what? C12. H12, I have what? 24, right? 24 minus 2, I have what? 22. Isn't that correct? Now, O6 plus O6 is what? O12. Then O12 minus this, you have what? O 11. So this is what? A disaccharide, because two monosaccharides have been reacted and water molecule is removed or eliminated from it. You now have what? A disaccharides. What about polysaccharides? Polysaccharides, we have example to be chitin, starch, cellulose. All of these are what? Poly. They will ask which of the following is a polysaccharide, or which of the following is not a polysaccharide. Starch, glycogen, chitin, cellulose are all polysaccharides. Sucrose, maltose, and lactose are all what? Disaccharides. Glucose, galactose, fructose, glyceraldehyde, arabinose, ribose are all what? Monosaccharides. So please, I said digestion and absorption of food molecules occurs in the ileum of the small intestine. The small intestine has villi, which provides large surface area for what? Absorption. Now, the function the trypsin have given us, it converts peptone to polypeptide. The ribosome convert polypeptide to what? Amino acid. Take note of that. Okay, now how do you test for starch? Let me quickly say this. Starch, which is a polysaccharide, is tested using what? Iodine solution. When starch is being tested using iodine solution, all right, coloration such as black to blue will appear. Now, black to blue PPT shows presence of what? Starch. So iodine solution is used to test for what? Starch. Black blue PPT shows the presence of what? Starch. Now, what about okay, Benedict's of Fehling's solution is used to test for what? Glucose. All right. Formation of brick red shows the presence of what? Glucose or fructose. Presence of brick red shows the presence of what? Glucose or what? Fructose. Benedict solution is used to test for glucose. Iodine is used to test for what? Starch. Okay. Now protein is tested using different solutions. All right. We have the Burette solution. When protein is tested using Burette solution, all right, purple or violet PPT is formed. When protein is tested using Burette solution, like Burette solution, you have what? Purple or violet PPT. Number two, you also have what is called a Millions test. All of these are tests for what? Protein. Now, when protein molecules are tested using this Millions solution, you have what is called what? Red PPT. Red PPT shows presence of what? Protein under what? Millions solution. Then purple or violet PPT shows the presence of protein under what? Burette solution. Now number three, you have the xanthoprotic solution. Now xanthoprotic solution, you have YL only. At first, you be have that is when you boil, that is we have white PPT, we have yellow PPT, and we have what? Orange PPT. All right, so when protein is being boiled, all right, it changes to white. Upon cooling, you have yellow. Then yellow, you now have what? Orange. So these are the three color changes that occur when protein substances are tested using what? The xanthoprotic what? Solution. JAMB, just taking of the color changes or the color of food molecules shown, or the solution, or the color of solution shown. All right, Burette is purple or violet, Millions is red, then Zato you have white, yellow, and what? Orange. Benedict is brick red, then starch is what? Iodine. Now, how do you now test for fat? Fat can be tested using translucent test, and Sudan 3 solution. All right, please. Formation of black PPT. Formation of black PPT shows the presence of what? Fat with Sudan 3 solution. When fat is tested using Sudan 3 solution, you have what? Black PPT. Take note of that. It is very, very important. So I just wanted to just give us this one. I oughtn't to have given it because I'm treating digestive system, but my mind just told me just say it. So I've said more than enough there. Starch is tested using iodine, so on and so forth. Now please let me give us this. Carbohydrates digestive enzyme is called what? Amylase. All right, carbohydrate digestive enzyme is called what? Amylase, and amylase converts starch to what? Glucose. Then number 2, you have protein. It is the type of bond present in carbohydrates is called what? A glycosidic bond. Glycosidic bond is present in carbohydrate. Carbohydrate digestive enzyme is called what? Amylase. Now, the type of bond present in protein is called what? A peptide bond. Peptide bond is present in protein. Protein has its digestive enzyme to be what? Protease, which convert protein to what? Amino acid. Number 3 is what? Lipids. Okay, let me use fat and oil. Yeah, for lipids. You know, lipid is the general name for both fat and oil, for both fat and what? Oil. So the digestive enzyme of fat and oil, or lipids, you say is called what? Lipase. Lipase converts fat and oil into fatty acid and what? Glycerol. Into fatty acid and glycerol. Please, carbohydrate digestive enzyme is called amylase, which converts starch to glucose. Protein digestive enzyme is called protease, which convert protein to what? Amino acid, which is the end products. Lipase is the digestive enzyme of lipids, of fat and oil, and it converts it to what? Fatty acid and glycerol. Take note of that. Please, in fungi such as mycorrhizopores, mushroom, bread mold, black bread mold, they store carbohydrates as glycogen, not as starch. Okay, now the cell wall of fungi is made up of chitin and not cellulose. It's made up of chitin and not what? Cellulose. Now, what kind of digestion do they undergo? Please, fungus or fungi undergo extracellular digestion. Extracellular what? Digestion. Then sponges also undergo extracellular what? Digestion. But hydra belong to class Hydrozoa. Other member of class Hydrozoa, you have Obelia and Hydra belong to class Hydrozoa. Now, digestion in Hydra occurs in the enteron. Please take notes. Digestion in Hydra occurs in what? The enteron. And in the enteron, you have extracellular digestion, while in the endodermis While in the endodermis, all right, you have what? Intracellular digestion. So which means that in Coelenterate or Nigerians, all right, there is hydra to be specific. It undergoes extracellular and what? Intracellular. Extracellular occurs where? In the enteron. While intra occurs what? In the endodermis. Now, what is the locomotory organ of hydra? It is what? Tentacle. The locomotory organ of hydra is called tentacle. The locomotory organ of hydra is called what? Tentacle. Now, the stingy cells with which hydra uses to trap its prey are called nematocysts. Now, they'll ask you which of these organisms possess stingy cells. It is what? Hydra. Hydra possesses stingy cells called nematocysts, which carries out the process of what? Feeding. It traps their prey. Nematocysts are found abundantly in what? The tentacles. Nematocysts are also found in the epidermis and endodermis, but they are abundantly found or distributed in the tentacle. Tentacle is meant for both feeding and movement. Okay, nematocyst is also meant for what? Feeding. Digestion is both extra and what? Intracellular. But a fungi, digestion is what? Extracellular, and this is done as a result of the presence of what? Hyphae. Hyphae produces an enzyme that helps in breaking down the substrates or the dead and decaying organic matters they are found growing on. All right, produce enzymes and they help in feeding on them. Take note of that. So at this point, okay, before I call it a day, and again, good, we now have accessory organs that carry out digestion. Now, the liver produces what? Bile. The liver produces bile, and the bile is stored in the gallbladder. Take note of that. Oh, the liver produces bile and is found stored in the gallbladder. The function of the bile is to emulsify fat. What? Fat. This red blood cell is destroyed in the liver spleen. Red blood cell is destroyed in the liver spleen through a process called erythroblastosis. The liver produces bile and bile is stored in the gallbladder, and the bile fat is what? Emulsified. I said red blood cell is destroyed in the liver spleen through a process called what? Erythroblastosis. All right. While the production of the red blood cell in the bone marrow is called what? Erythropoiesis. So which means that the reverse of erythroblastosis is what? Erythropoiesis. Production. Please, in babies, red blood cells are produced in the liver because they don't have what? They don't have a well-formed bone. So their liver produces what? Red blood cell. But in adults, it is produced in the bone marrow. So, okay, let me do this. I'm running off with this. I have something like this. Please take note of what I'm drawing now. So I have let me do a sketch of. All right, so I have just I'm drawing it in a hurry. That's why it's like this. So this is just the structure of what? The digestive system. All right, of course, this place is the esophagus. All right, this first region I said is what? The cardiac sphincter. All right, and of course, this is the stomach, and this is the liver, and inside of this structure is where you have the bile, and the outside this you now have what is called the gallbladder. So which means that the contents of the gallbladder, just as I said, is what? The bile. Okay, and this place is called what? The bile duct. Bile duct. So this place is called pancreas. Pancreas. Now this region here, this region here, this very region is called the duodenum. Duodenum. And of course here is called the rectum, and this small hole is called what? The anus. Alright, this is small intestine inside, and this outer one is called what? The large intestine. So I've explained everything here. Now this lower region of the stomach I told us is what? The pyloric sphincter. Alright, pyloric was sphincter. So at this point we call it a day. Thanks for watching. Do have a wonderful day.